All right. Good evening, everyone. My name is Xin, and this is Erika. And it is truly an honor for both of us to be here tonight. And although both of us has moved on from MIT and started our own career, and so I'm currently assistant professor at K-State, and also Erika already in the process of starting her own new lab. We are especially grateful for this chance to reconnect with the KI communities and also MIT community, and to meet with so many old friends and our colleagues. And we would like to begin by sincerely thanking the MIT Image Award Committee for recognizing our work, and we are very excited to share not only this image, but also the biomedical idea behind it. And today we are going to presenting this work, which is the Jeweled Scaffold, a programmable drug-eluting stent, a platform designed to support the healings after a heart attack through localized and programmable drug delivery. When people think about the life-threatening disease, a cancer often comes to the mind first. But globally, the cardiovascular disease, remains the leading cause of the death. The heart disease causes nearly 9 million deaths every year, accounting for about 1 in 6 deaths worldwide. And a heart attack happens when a coronary artery becomes blocked, cutting off the blood supply to the heart. In the United States, this occurred about every 40 seconds, and even when we successfully reopen this artery, many patients still go on to develop heart failures because the injured heart tissue cannot fully recover. One of the most common treatments for heart attack is the coronary stent, a small mesh tube that can reopen the blockage and then restore the blood flow. And during this process, a catheter carries a collapsed stent to the site of the blockage where a balloon is bent against the vessel wall. Stents are actually highly effective at the fixing of the blockage, but they do not address the biological damage. And that will continue to unfold the heart after blood flow is restored. This is actually the gap we are going to address with this idea.
So after the initial injury, the heart actually goes through a series of stages, and ideally it will require different therapeutic interventions at different times. And staged and localized therapies have been extensively discussed in the cardiac space. But until now, the technology to achieve this level of control hasn't been really available. So this really motivated us to develop timed temporal intervention with microparticle delivery and encapsulation and delivery system. This is an implantable patch that is capable of releasing different cardiac regenerating agents in a fully programmable sequences for heart attack. And building on this work, we started to wonder whether the timed system can be directly integrated into a coronary stent, so we could deliver post-MI therapy in a non-invasive manner, right when the artery is just reopened. So to do that, we designed this 3D lattice scaffold that forms the stent body. And we 3D printed this prototype and integrated this tiny pockets across the stent body, so they are not really interfering with the radial expansion. And we then use a PLGA, an FDA-approved polymer, to fabricate these microparticles with this tiny central reservoir, which can be then loaded with different therapeutic compounds. And each of these particles can be sealed with a cap, which then degrades over time, enabling pulsatile drug release. And by tuning the material properties of these caps, we can precisely control timing of each release event. And finally, We color code each particles to mark different release timing, and we specially arrange them into these pockets across the stent body, creating this jewel-like structure you see in our image, where each gemstone represents an individual delivery event. And eventually, this really comes down to whether our device can actively guide how heart generates. So building on our timed work, we observed a pretty remarkable 50% reduction in infarct size, as well as a twofold increase in neovascularization, as you can see from our the right stent blood vessels, all suggesting our device can really meaningfully reshape how heart regenerates after a heart attack. And the same therapeutic program can be directly encoded into our stent technology. And we expect to see further enhanced tissue regeneration and restored function, even without invasive surgical implantation or reverse remodeling. So as a quick summary, in this image, we present a proof of concept prototype of a specially arranged phase-specific drug delivery stents, which moves beyond the conventional drug-eluting stents, which typically deliver one drug in a burst. And moving forward, we are working to fine-tune the release timing and different therapeutic combinations to develop more clinically aligned treatment for IMI or heart attack. And we're also exploring a scalable fabrication process, such as direct printing on metal, such as nitrile, which is a clinical used flexible and self-expanding material. And ultimately, our goal is to translate this technology into clinically deployable method and transforming stent from a passive implant into an active therapeutic platform. And with that, we'd like to thank Anna and Bob for their incredible support. And our collaborator, Dr. Chen from Columbia University, who spent some time visiting the Langer Lab last year. This work really grew out of a wonderful collaboration. And lastly, we'd like to thank everyone in the Langer Lab and Jack Lannon group for their support along the way. And lastly, I'd like to give a special shout out to my daughter, Kira, as I took this image with Xing when I was 6 months pregnant. So she's also part of our team, technically. And lastly, thank you so much, everyone, for your time. And thank you for selecting our image.